get started. So hi everyone, welcome to this week's Tuesday lunch seminar, where we invite internal speakers from our EEB department to talk about their work and research. So today we have Dr. Joseph Charboneau, postdoctoral scholar in the EEB department, working with Professor Stephen Smith. Joey received his undergraduate degree in biology and environmental studies with a minor in geology from Oberlin College in 2008. After some time off, he received his master's in botany from the University of Wyoming in 2013, where he worked with Dr. Ronald Hartman, and he went on to receive his PhD in ecology and evolutionary biology at the University of Arizona in 2020, where he worked with Dr. Michael Sanderson. And he continued there as a G.G. Simpson postdoctoral fellow until 2022 when he came to Michigan. Joey's research centers on plant systematics, phylogenomics, and biodiversity informatics, and has spanned a diverse array of projects ranging from phylogenetic methodology to empirical botanical research and contributions to digital herbaria management at Arizona. Joey has received many research grants and awards for his various work, including the NSF Doctoral Dissertation Improvement Grant and the IGERT Fellowship in Comparative Genomics from NSF, and has given numerous presentations at both universities and conferences. So today we will hear from him on character evolution and homoplasy in the New World milkvetches of the Astragalus and Fabaceae.
Okay, thanks, Avi. Thanks for coming, everyone. I'm excited to share with you some of the dissertation research I did in Mike Sanderson's lab at Arizona. Okay. Seems that it's frozen up a little bit. Maybe you don't have to be. There we go. Okay, so to take a very broad entry into this topic of homoplasy, I want to first introduce these two processes that were at play in evolutionary radiation, during which many species have formed over short periods of geologic time. So there's diversification, or how rapidly new species and lineages have originated, and disparification, the extent of differentiation among these species in traits is related to their different ecological niches. So when angiosperms quickly came to dominate most terrestrial ecosystems beginning in the Cretaceous, rapid diversification was accompanied by great disparification. So we can see that some of these disparate traits in the Sonoran Desert ecosystem that you can find at the foot of the Santa Catalina Mountains outside of Tucson in Arizona. And there's lots of different angiosperms out there, but I want to draw your attention to two of them. These saguaro cacti and this drought deciduous ocotillo, which has these orange reddish flowers right here. So these two have different forms that are related to their strategy of dealing with the challenge of lack of water. And the disparification of angiosperms has also led to the replication of similar forms in this giant succulent euphorbia from southern Africa and this drought deciduous alawadia from Madagascar. So we call this replication of the same forms convergent evolution or homoplasy. So homoplasy has also happened on shorter evolutionary timescales in recent rapid radiations, which are radiations involving rapid diversification accompanied by great disparification. So a great example of these are these Caribbean anoles, which have recurrently filled these different niches on different islands of the Greater Antilles and converged on morphologies that are related to those niches. However, sometimes there's been groups that have had high diversification rates but without disparification to match. So these are sometimes referred to as non-adaptive radiations. So species might resemble each other not because they have converged on new niches, but because they've retained their ancestral niches. So a possible example is a clade of plants called Astragalus in the Americas that has hundreds of species of predominantly temperate herbaceous perennials. So they show less disparity in growth form and climatic niche than classic examples of plant adaptive radiations like the Hawaiian silver swords. So often the focus of studies on large radiations has been on diversification rates, and less often has disparity been investigated in depth in groups like Astragalus. And perhaps there are more differences in Astragalus species than apparent at first glance. So these 7 Astragalus species look pretty similar superficially, but they do have differences that you can't see in photographs. So these 5 are specialists on different kinds of geologic substrates. And then these three produce or accumulate different kinds of toxic compounds to deter herbivores. And so at the center of these two cryptic or difficult to observe traits are about 24 Neo-Astragalus species that are so-called selenium hyperaccumulators. So these are not only restricted to soils that are already high in selenium, but they concentrate the element in their tissues to the point that they're highly toxic to herbivores. So potential homoplasy in a trait like selenium hyperaccumulation could be indicative of broader cryptic disparification in Neo-Astragalus. And so a question that motivates my research is, are non-adaptive radiations merely adaptive radiations involving cryptic or poorly known traits? And I've addressed this by examining character evolution and homoplasy in Neo-Astragalus and the selenium hyperaccumulation trait. And I've also examined the evolution of different morphological characters with regard to a morphology-based taxonomic system for the group. And I'll talk about morphology after I give you a little bit more background on Astragalus, and then I'll return to that selenium hyperaccumulation trait. So Neo-Astragalus is a clade in the genus Astragalus in the legume family, and these Astragalus species are commonly known as milkvetches or loco weeds, although the latter name is also applied to species in a related genus, Oxytropus. And a lot of people are familiar with the genus because of this species, Astragalus membranaceus, which is used in Chinese traditional medicine. What you may not know is that there are more species in the genus Astragalus than in nearly or perhaps all other genera of sea plants, with just about 3,000 species worldwide. And the genus is estimated to have originated only in the last 11 to 17 million years or so. So Astragalus species are my clicker is getting mixed up. Found mostly in the northern hemisphere in temperate regions and sporadically into tropical montane areas. And they are most commonly found in arid or semi-arid habitats. So most of the species richness is actually found in the Old World, especially in southwestern and central Asia. But there are centers of diversity in both North and South America, which together have about 500 species, 400 or so in North America and about 100 in South America. So of those 500 or so Astragalus species in the Americas, all but about 15 from North America are part of the Neo-Astragalus clade, which forms a natural group within the genus by its aneuploid chromosome series that has counts that are intermediate between the diploid and tetraploid counts for the genus, which has a base number of 8. And so Neo-Astragalus is estimated to have originated only about 4.5 million years ago. So it has a very high net diversification rate. But as I mentioned before, almost all Neo-Astragalus species are temperate herbaceous perennials. So to give you an idea of how great the species richness of Neo-Astragalus is in North America, here's a map of the density of native Astragalus species in the conterminous US. So the greatest density of species is found in Garfield County, Utah. So blue is more, and there are 59 species in Garfield County. There's over 100 in Arizona, which makes it the most species-rich genus in the flora, and that's true of a lot of other western states. There's only 2 native species to Michigan, including this one, Astragalus neglectus, which is in the Neo-Astragalus clade. The other one is not. But Neo-Astragalus species are more disparate in morphological traits than the photos I've been showing you so far suggest. So there's lots of variation in vegetative characters, like the form of the leaflets of their compound leaves, as well as variation in flower size and color and shape, although they have prototypically papilionaceous flowers. But Neo-Astragalus and all Astragalus species really are especially diverse in fruit or pod characters. So there's variation not only in size and shape and texture, but whether they are deciduous or persistent and how they open or don't to release the seeds inside. So to study character evolution in this clade, we need a well-resolved phylogeny. But clades resulting from rapid radiations are notoriously difficult to estimate phylogenies in. So the resolution of Neo-Astragalus gene trees in previous studies has really been hampered by a lack of sequence variation. So here's a phylogeny using ITS from a previous study. And Astragalus is in this gigantic polytomy nested within the rest of Astragalus, which is maybe not surprising given how many species there are and how recently they're supposed to have shared a common ancestor. But Astragalus as a clade has had strong support in subsequent studies, but the only subclades that have had any support to speak of really are these two here labeled F and G that contain South American species that are apparently the result of two long-distance dispersal events. So the genome skimming sequencing that I have done has provided orders of magnitude more characters for chloroplasts and nuclear ribosomal DNA loci to estimate and improve phylogeny. But with hundreds of species, I had to choose which ones I was going to sample for the phylogeny, and for that I turned to Rupert Barnaby's taxonomic system for the genus in North America. I guess I've given this talk before online a lot, and so I could just pick up the books that I had sitting next to me, but they're so big I didn't want to bring them in. It's in 2 volumes, and it's like that many pages. It's really big. Monumental is always the adjective that people use to describe it. But Barnaby divided up the almost 400 aneuploid North American species into 3 informal, what he called phalanxes, and 83 sections, which is a infrageneric taxon rank. And this was on the basis of different morphological characters. And he attempted to form a natural taxonomic system that's meant to reflect the phylogenetic relationships among species by considering the possible paths of evolution that he saw in these morphological characters, while recognizing that homoplasy must have taken place. So with the most recent taxonomic revision by Stanley Welsh, there are now 85 aneuploid sections, and those South American species were placed in 2 informal groups by Ivan Johnson in the 1930s and 40s. And these have largely corresponded to the 2 clades that include South American species that have been identified in previous work. So I made a lot of collections to try to sample as broadly taxonomically across these aneuploid Astragalus sections as possible. And I've made over 500 total collections now of 114 different Astragalus species. If you include these collections I made in northeastern Montana, which I did as a master's student at the University of Wyoming, and that's actually how I got interested in Astragalus in the first place. So I also want to add that I rounded out the sampling with herbarium specimens. About a third of my samples came from existing herbarium specimens where the curators gave me permission to sample. So I ended up including 94 Astragalus species and 90 Neo-Astragalus species among these samples, and there are representatives of 55 of 85 aneuploid sections, including all those that have at least 3 species. Plus, I also sampled all 26 species that are thought to hyperaccumulate selenium. So I started with relatively high-depth Illumina HiSeq data from 3 Neo-Astragalus species. The data from one of those came from my collaborator, Aaron Liston at Oregon State University. And I did some de novo assemblies of those to get in-group references for the plastid genome and nuclear DNA. For most of the samples, I prepared libraries for NextSeq sequencing at Rich Cron's lab at the U.S. Forest Service Research Station in Corvallis, which is where Oregon State is. And I used maps from each of these samples I used reads from each of these samples and maps to the in-group references and to do de novo assembly of both the plastome and the nuclear ribosomal DNA. And just a quick aside about Astragalus chloroplast genomes. It's part of a large clade of legumes called the inverted repeat lacking clade. So this inverted repeat, or IR, is a large feature of the chloroplast genomes of almost all green plants that's missing from this particular clade. It's not the only one where that feature has been lost, but it's the most species-rich group. But something that seems to be associated with the loss of this inverted repeat is frequent large-scale plastome inversions. So almost 20% of all the samples I had at least one of these inversions, which is what this mess of a diagram is indicating. So this is in alignment with this program called MOVE that identifies locally collinear blocks, which is what each of these colored pieces are, and where they change sides on this horizontal axis is where there's been an inversion. But basically, by doing this alignment, I was able to include intergenic spacers in my phylogenies, in addition to just the coding sequences, because those coding sequences don't have nearly as many variable characters as the stuff that's not genic. So let's take a look at some of these phylogenies. This first one is the plastome tree. So it's a majority rule consensus cladogram from 1,000 ultrafast bootstrap replicates, estimated with IQ tree using maximum likelihood. And so these dark blue nodes are next to branches with 100% bootstrap support. And there's quite a few of those in this plastome phylogeny. And we can compare this to the nucleoribosomal DNA phylogeny, which still has quite a few well-supported nodes, certainly more than like that ITS tree from back then did that I showed you earlier, but not nearly to the same extent as the plastome tree. But both of these have Neo-Astragalus forming a clade with 100% bootstrap support, which is in agreement with previous studies. And these dashed lines that I'm showing you now connect the taxa in the two different trees. So I think there's actually quite a bit of concordance between them, though obviously there are some taxa that are showing up in very different places in each of those phylogenies. So one place where they agree is this clade of 5 species that is sister to the rest of Neo-Astragalus, that I've called the Pan-American clade. And that's because it contains species from the southwestern US, central and southern Mexico, and then barely into Central America, and then also species from South America. So one of the species from the southwestern US is Astragalus natalianus, which you can find in people's yards growing in Tucson. And another clade that's common to both is what I'm calling the Pacific inflati clade, that includes species from South America, as well as many from California and Baja California, like this Astragalus natalii from the California coast. And that these two species are Astragalus natalianus, and natalii, I think, is indicative of just how many names there have been published in the genus. So with these improved phylogenies, we can evaluate Barnaby's taxonomic system for its correspondence to the trees. And I'm going to talk just a little bit about a couple of morphological characters that he considered most important in forming his system. So the first is stipule connation. So stipules are these leaf appendages that come in pairs that you might be familiar with from acacias or mesquites, some of which have them modified into spines. In Astragalus, they're not spinescent, but in some species they are connate or fused together, and in others they remain distinct from each other. And so a second important character for Barnaby's system was whether or not the pods were persistent or deciduous. So in some species, the pods are deciduous from the plant before they shed their seeds. If you look here, there's a pile of these pods on the ground. And in others, they remain on the plant until after they've shed their seeds. And pod division was a character that Barnaby recognized to have considerable homoplasy, and it little informed his taxonomic system. So some species have unilocular pods, and others have bilocular pods, with the two locules separated by a septum, like this one. And he didn't weight this character very heavily because there are even varieties of the same species that differ in it, and so he used it really only to differentiate among closely related species. So I encoded these characters and some others as binary characters and did ancestral state reconstruction using stochastic character mapping to calculate the probability of each state along the internal branches of the tree. So here I'll just show some examples of what the evolution of characters with varying levels of homoplasy might look like in these different reconstructions. So this first one has two states, A and B, shown in blue and red. The colors in A and B are in no way meant to indicate the ancestral state. But there's no homoplasy that's taken place in this. There's just been one transition from one state to the other. So that's kind of like the dream to a taxonomist, I guess, is that there's one feature that can distinguish between, that can identify at least a single clade. So if homoplasy is present, it could look potentially like this, where the second character has some homoplastic occurrences of state B, but they mostly occur toward the tips of the trees. And homoplasy can also be very predominant. So this third character has several transitions between character states throughout the tree. So homoplasy in these morphological traits that I considered was pretty rampant, like that ladder tree from the previous slide. So these are the state reconstructions for stipule conation in the plastome and ribosomal DNA trees. And you can see that there's been quite a bit of transitions back and forth between connate and distinct stipules. So the same is true for the pod persistence characters and the pod division characters. And when we look at the estimated transition rates and the fewest possible changes between states, for these three characters, I think it's interesting that the two that Barnaby weighted so heavily, stipule connation and pod persistence, have just as much, if not more, homoplasy than that pod division character that he thought was only useful for distinguishing between closely related species. So from these results, we can see it would have been pretty much impossible for anyone to create a natural phylogenetic classification system based off of just a small number of morphological characters. But the flip side of this, I think, is that this high levels of homoplasy mean that even closely related species might differ in their character states, and in practice can be differentiated from each other, which has been, at least my experience. Anyone who's tried to key out one of these species may disagree with me, though. There's also been homoplasy in ecologically important morphological traits like the deciduousness of the pods, which is important in determining how far the seeds are dispersed, but also in traits I haven't mentioned, like those related to pollination. So here's a classic hummingbird pollination syndrome that has apparently originated probably 3 times in the genus independently. Okay, so to transition back to this selenium hyperaccumulation trait, I'm going to take you back to chemistry class here. Selenium is element number 34. It's in the same group as oxygen and sulfur. And high levels of selenium are toxic to most organisms. It's actually a required micronutrient for all animals, including humans though, but plants don't seem to require it. Selenium becomes toxic when it replaces sulfur and sulfur-containing amino acids, methionine and especially cysteine, which forms disulfide bridges that stabilize protein conformation. So these cattle require selenium in their diets, which they get from the plants that they eat that in turn got it from the soil that they're growing in. But too much selenium is toxic to both the plants and the animals. And there's actually lots of livestock that is lost each year through chronic and acute selenium poisoning, usually in areas with high soil selenium concentrations, which are those indicated in pink on this map of the western US. So some plants are tolerant of these high selenium concentrations in the soil, and some even concentrated in their tissues. So the plants that accumulate the highest concentrations of selenium we call selenium hyperaccumulators, which often concentrate it by over 1,000-fold relative to the soil that they grow in. So there are other plants that hyperaccumulate trace elements. For example, many serpentine-adapted plants hyperaccumulate nickel. But for all elemental hyperaccumulators, there are different statuses or levels that are applied to taxa, which have threshold criteria for classification. But the threshold concentration is different for each of these elements. So for selenium, non-accumulators are taxa that have individuals assayed with less than 100 milligrams per kilogram dry weight selenium, which is equivalent to parts per million. And accumulators are taxa with individuals having between 100 and 1,000 milligrams per kilogram selenium. And hyperaccumulators are taxa with individuals having more than 1,000 milligrams per kilogram. Some of these can even exceed to 10,000 milligrams per kilogram, which is equivalent to 1% dry weight. And for convenience, I'm just going to refer to plants and taxa that are either selenium accumulators or hyperaccumulators as selenophytes. So I want to make it clear that one of these statuses is applied to a species or a variety if any individual anywhere at any time has ever met that threshold criterion. And so selenium hyperaccumulators are known in 14 different genera and 6 flowering plant families. And the 3 families that have the most species are Astragalus, Brassicaceae, which has this well-studied genus Stanleya, and of course Fabaceae, which has more selenium hyperaccumulators than any other family, most of which are Astragalus species. So, as I mentioned, there's other plants that hyperaccumulate metals, and the hypothesis for why they do this is called the elemental defense hypothesis. That basically you accumulate stuff from the soil to the point that anything that tries to eat you is poisoned. So there's this whole world of ecological implications for hyperaccumulation of selenium that's pretty fascinating, I think, but that I won't really have time to get into here. So the 26 Neo-Astragalus species thought to be selenophytes are found in 2 groups in Barnaby's taxonomic system, which actually differ in that stipule connation character. So there are 16 species with connate stipules. He placed in 6 different sections within a group he informally called the Seleniferous homolobi, and 10 species with distinct stipules that he placed in a single section, Prussiani. And so Barnaby thought that these 2 groups were not closely related at all, so this provides us with the hypothesis of 2 independent origins of selenium hyperaccumulation. But the monophyly of these 2 groups has not been confirmed by previous molecular work. So you may be asking, how does one identify a selenophyte? So most of the time it's actually been from the way that they smell in the field. So they emit this pretty distinct metallic garlicky odor is how I would describe it, that is from these selenium-containing volatile compounds that the plants release. Most people think that the smell is pretty unpleasant. I actually kind of like it a little bit. I thought about having like a pass around the thing and smell it, but since people are eating and people don't like it, it didn't seem like a good idea. And I just want to read this passage from Barnaby's atlas because I think it really encapsulates what was known or assumed about the ability of some of these taxa to accumulate selenium. So he is writing about Marcus E. Jones, who had written a previous taxonomic treatment of North American Astragalus. Jones described the plants as having a strong snake-like odor, a term which he used repeatedly for the malodorous selenium-converting species. Well, this was also true of the material that I collected. Well, I cannot be sure without chemical experiment that Astragalus woodruffii is seleniferous. There is presumptive evidence that such is the case, and this is in harmony with what can be assumed from exterior morphology about the probable affinities of the species. So there have been chemical assays to quantify the selenium content of plants since the 1930s. So a bunch of these have been confirmed as hyperaccumulators, but a lot like this Astragalus woodruffii have not ever been assayed for selenium content. So here are photos of each of the taxa, including varieties of these 2 groups, the Seleniferous homolobi and section Prussiani, which collectively I'll call the putative selenophytes. And these 4 species along the bottom are potentially selenophytes known to grow on selenium-rich soils, but don't smell strongly. And so these are also potentially thought to be closely related somewhat to section Prussiani. And then here's a representation of what was known about the selenium accumulation status of these taxa. So some had been confirmed with assays as accumulators or hyperaccumulators, but there are many others that were just assumed to be selenophytes based off of the way that they smell. So I sought to confirm the status of these selenophytes by conducting my own selenium assays with this technique called ICP-OES, which I did at Elizabeth Peelan Smith's lab at Colorado State. And so when I was out collecting plants to extract DNA from, I was also collecting leaf samples to do the assays on 3 individuals per population. And I also collected soil samples as well from each of those locations in pink on the map. So I've now done assays on at least one population of 25 of 26 species and 37 of 39 putative selenophyte taxa, as well as all 4 of the potential selenophytes, and a few others that have never been thought to be selenophytes. So as a result of my assays, as well as previous studies, nearly all putative selenophyte taxa have now been documented with at least accumulator concentrations of selenium. So in these figures I'm showing on a log scale the maximum leaf concentration recorded from an individual in each taxon. So anything with greater than 1,000 parts per million, or 10 to the third, is classified as a hyperaccumulator. Anything from 100 to 1,000 is an accumulator. And then there are a few that have been classified just as non-accumulators with less than 100 parts per million. So what this doesn't show is the variation that exists among individuals in the same taxon. So remember that the accumulation levels are assigned to taxa based off of one individual ever having met that threshold concentration. And not every individual assayed is going to have that selenium hyperaccumulation concentration of selenium, even if that taxon has been classified that way. So this black horizontal line that I've added indicates the range of concentrations ever recorded for a taxon, both in mine and in previous assays. And for some of these, they can span almost 4 orders of magnitude. So there's even a lot of variation in selenium concentrations among individuals in the same population, which I'll illustrate with just these 2 species. So each of these lines represents one population and the 3 different individuals I sampled from it for selenium concentration, so each of the 3 points on one of those lines. So you can see that a lot of them differ by an order of magnitude, and they often differ on which side of those thresholds they're on. And in extreme cases, individuals from the same population might differ by even almost 2 orders of magnitude, like this one right here. So given the variation in selenium concentrations we see within taxa and populations, it may be necessary to sample quite a few individuals from a taxon that's capable of hyperaccumulating selenium before you actually encounter one with a hyperaccumulator concentration. So even after my sampling, there's still several taxa that have only ever had 3 individuals assayed. So because of the similar biochemical properties of selenium and sulfur, many proteins that act on sulfur-containing metabolites are perfectly capable of acting on selenium analogs as well, but usually a lot less efficiently. And that's how selenium gets into most plants in the first place, as a byproduct of them actively transporting sulfur as sulfate into their roots. So selenium hyperaccumulators then must have some way of discriminating between selenium and sulfur-containing metabolites, because they're able to concentrate selenium to a much greater extent than they are to concentrating sulfur. So we can get a better indication of an individual's ability to accumulate selenium by examining selenium concentrations relative to sulfur concentrations in their leaves, and that ratio also relative to the soil that they're grown in. So I collected soil samples from the field and assayed them for, let's see, so I collected soil samples from the field and assayed them with this other technique that's more sensitive because the concentration of selenium is much lower in the soil, to look at how these ratios of selenium to sulfur have changed, going from soil to plant. So to illustrate what you can get from these selenium and sulfur ratios, I'll show you some examples. So here I'm showing the log of selenium and sulfur concentrations on the y and x axes. And a single point on this graph represents the selenium and sulfur concentrations of an assayed sample. So here's one potentially for a soil sample. And we can plot 2 points for an individual plant, one for the soil and then one for the plant itself. And you can connect these 2 lines that indicates the difference between those. So in the case of this particular plant, it concentrated selenium and sulfur to an equal extent by 2 orders of magnitude each. And so this arrow has a slope of 1 on the graph. So here is an arrow for a different individual growing in a different soil. And in this case, the plant has concentrated sulfur to a much greater extent than it has selenium. And so it has a slope of this line of less than 1. So this is what we expect from non-accumulator plants. And now here's a third arrow for a plant that's concentrating selenium to a much greater extent than sulfur. And so it has a slope of greater than 1. This is what we expect selenium hyperaccumulators to have. So when we look at these arrows that indicate these changes from soil to leaf, you can start to see differences between these different statuses and what the plants are doing with selenium versus sulfur. So most individuals from non-accumulator taxa are concentrating sulfur from the soil without concentrating selenium to the same extent. So they have these lines that have a slope of less than 1. There's a couple that obviously have a higher slope, greater than 1. Those are ones that never got with the accumulator concentration, but they're probably capable of doing it if we sample more of those individuals. And the individuals from accumulator and hyperaccumulator taxa are concentrating selenium quite a bit more than they are sulfur. And they're not necessarily doing it to that much greater of an extent as the hyperaccumulator taxa compared to the accumulators. But now that I've talked about how we identify these selenium hyperaccumulators, let's look at where the putative selenophytes fall out on this phylogeny. And these are the same plastome and NR-DNA trees that I showed earlier. One of these 2 hypothesized groups of selenophytes, section prussiani, which is shown here in pink, forms a paraphyletic group in both trees. And the other hypothesized group, the Seleniferous homolobi, which included 6 different sections, is not monophyletic in either tree. And species in, let's see, 2 other sections of the Seleniferous homolobi, Pectinati and Woodruffiani, form a clade in the ribosomal tree, but they're in 2 different clades in the plastome tree. That's this group in green here. So I performed ancestral state reconstructions of both as a discrete and continuous character of selenium accumulation. And as a discrete character, I assigned an accumulation status to each species in the tree according to those strict threshold concentrations. And I did ancestral character state reconstruction using a model that allowed transitions between all 3 states with symmetrical rates. And so these circles are the probability of each of those states at that particular node. So I also encoded the maximum selenium concentration on a log scale to do continuous character reconstruction using a Brownian motion model. So the color that is along each of these branches represents a maximum selenium concentration that might have been found. If I didn't have data for a species, I just coded it as having a maximum selenium concentration of 1 and as a non-accumulator, which I think is not that unreasonable of an assumption because these things really do smell. And humans are pretty good at detecting it. So if there were some that we didn't know about, we probably would be able to tell just by smelling them. So another thing is this tree makes it look like almost half of the species in Astragalus are selenium hyperaccumulators, but you gotta remember that it's biased toward including those in the phylogeny. So if we zoom in on just this part of the tree, we can look at this a little bit better. And it looks like there have been 4 to 5 independent origins of selenium hyperaccumulation from the plastome tree. So it's really hard to see the screen from over here, but 1, 2, I don't know. I can't really see it. Hopefully you can see that, or trust me on it. So in these, we also see that selenium hyperaccumulation is inferred to have originated from a non-accumulator ancestor. That's not really what we see in the continuous reconstruction, but I think that's probably because the Brownian motion model is not the best model to use in thinking about these concentrations. So we can do the same thing for the ribosomal DNA tree, and I'll do the same thing and just zoom in on this a little bit to see it better. And there appear to have been 3 origins of selenium hyperaccumulation from this in our DNA tree, but they're not, they don't correspond with the same origins in that plastome tree. So there also appear to be some losses of this trait, and I think that one of these is actually capable of accumulating selenium. That's the one that had the anomalous slope on that graph a few slides ago. One of them I think really has lost the ability, and another is just one that is unexpectedly related to these selenophytes that nobody thought was a close relative before, and so nobody's assayed it. But it doesn't smell strongly. So what its true status is really remains to be seen. So it looks like there have been between 3 and 5 independent origins of selenium hyperaccumulation in Astragalus, which is actually even more than Barnaby had hypothesized. But the discordance between these 2 trees makes the number and placement of those origins less certain. So I think it's actually pretty remarkable that there appear to have been at least 3 origins of this trait in just this one clade, because it's something that's only known, selenium hyperaccumulation, in 13 other genera of flowering plants. So homoplasy in morphological characters has occurred in Astragalus, as well as these multiple origins, apparently, of selenium hyperaccumulation. And cryptic disparity in homoplasy and ecologically important traits like specialization on selenium-rich soils and selenium hyperaccumulation may be indicative of broader disparification, I think, that has occurred in Astragalus. And so these improved phylogenies provide a foundation for future studies in the group. And I think there's some really interesting stuff going on with whole genome duplication and chromosome number evolution that have given rise to that aneuploid chromosome series in the group. So I just wanted to briefly mention the project I'm actually working on here at Michigan, which is this phylogenomics project on African acacias. So they're also in the legume family, which isn't that big of a leap for me. But there's about 130 African species of these trees that are in 2 segregate genera called Vechelia and Senegalia. The taxonomic history of acacia in the broad sense is really complicated. But I'm working on the phylogenomic aspects of this project, but there's also collaborators we have at Wake Forest University who are focusing on some of the functional genomic and ecological components of the project. The idea is to try to understand the origins and genetic basis for several traits that are related to living in these open savanna ecosystems, like that they have to deal with fires and competition with grasses and large herbivores, and it's pretty dry there as well. So I want to acknowledge members of the Sanderson Lab at Arizona and the members of my dissertation committee. My collaborators have been a big help in this as well. I mentioned Aaron and Rich and Elizabeth, and I've also been working with Marty Wojciechowski with this at Arizona State. And I received a lot of guidance from the folks in those other labs, and I also want to acknowledge a couple of undergraduates, Lena Nguyen and Wesley Wilcoxon, who helped me at Arizona, and Sanjana Nagamangala, who is a undergrad who started helping me in the lab this semester. So I worked with a lot of different places to secure collecting permits, and the staff at those herbaria allowed me to sample destructively, so I thank them. And thank you for coming and listening, and I'm happy to answer any questions that you have. Oh, definitely there's a lot of variation, and that probably has a lot to do with the variation you see in just like that simple one sample assay. So some evidence for that elemental defense hypothesis are that you find the highest concentrations in the most reproductively important parts of the plant. So fruits and flowers tend to have higher concentrations, as well as the newest leaves. So yes, it's pretty variable.
And related to that, do you see any evidence of recovery on any of the humidity in the cells in the field?
Yes, so this is another part of the whole world of ecology of these that I didn't have time to get into. There are insect herbivores that have developed their own resistance to the selenium levels. So there definitely are herbivores on them. Yes.
Do you find those selenium accumulators in certain habitats, like certain food soils or in certain metal?
Yes, so they are found like almost exclusively in soils derived from particular geologic formations that are high in selenium.
Those tend to be pretty locally endemic.
Yes.
Okay.
Yeah, so a lot of these are like on the Colorado Plateau. And so you mostly find them on like sedimentary formations, like sandstone and shales. And so just different pockets of where those are exposed might have a different endemic species on them. Yes.
I'm just curious, what's the evidence, or is there evidence for reproductive isolation between these different groups? Like, if we know for sure that they're good species, or is it possible that they're just locally adapted populations with the same reproductively non-isolated?
Yeah. I don't have a good answer to that based off of the data that I've collected. There are several varieties of some of these, and some of them have pretty widespread geographic ranges as well. I have libraries for some other like varieties of these taxa and like multiple individuals from some populations, but I don't have the sequence data from those yet.
It'd be interesting to look at, like, just even the scale of FSTD as a really coarse measure of divergence.
Mm-hmm.
To get a sense if it's like, oh, this was the real, it's just interesting seeing that polytomy and then the trouble that you had with reconciling the two different phylogenies you got
Mm-hmm.
quickly because it's just really shallow differences.
Yeah, definitely. I guess something that makes me hope that they are distinct is just that they do really have distinct like morphological differences between them. But we'll see. Yes.
Yeah, so given that you found that this was like a very rapid radiation and that some of these traits are polymorphic even within individual species, is it possible that some of these transitions are not homoplasy transitions but actually represent like hemiplasy?
Mm-hmm.
That they're like ancestrally polymorphic
Right.
and sorted differentially?
Right. Yeah, I think definitely that's a possibility. So another aspect of this research, I've collected target capture data for phylogenomics, but also targeted some functional gene families that are related to selenium and sulfur metabolism. So kind of elucidating what the actual molecular mechanisms are, I think will be enlightening, and there's definitely like multiple copies of some of these genes in a gene family. So I think it's going to be an interesting story. Bobby.
We have 3 questions in the chat. So the first is from Catherine Badley. Is there any geographic relationship between the geographic distribution of the selenium accumulators in bedrock geology?
Yeah, I think that's what Chris was kind of getting after. But there are some pretty interesting geographic patterns that have to do with like the continental divide. So there's like one of those groups, section Pectinatae, it had like 2 clades in the plastome tree. And those two clades, like you find species on one side of the continental divide and the other on the other. And this other genus, Stanleya in the mustard family, it really only has one taxon that has been found to have the hyperaccumulator concentrations of selenium, and it's only populations on the east side of the continental divide also that those have been found in.
So let me, maybe I can briefly clarify too, or add a little more to my question.
Okay.
Thanks for an interesting talk. If you were to show a map of the selenium-rich bedrock formations, how many of those would be occupied by Astragalus species, and how are they dispersed? Are they dispersal limited in some way, or would you find that they can easily disperse to other selenium-rich sites if they're going to do well there?
Yeah, I think they are dispersal limited. They just form these pods, a lot of which are dry, and will shed them or not. I think some species there have pods that are allowing for more wind dispersal, like some of them will split open, or they're really pretty porous, so they get blown around by the wind. But dispersal from one patch of the selenium-rich soil to another is definitely something that's probably not happening all that often.
Thanks.
And we have another few questions from David Meisner. Great presentation. How mobile is selenium in the soil/root interface? And is there microsite variation in selenium levels that might account for interpopulational variation in accumulation?
Yeah, one of the students who graduated from Leanne's Peelan-Smith's lab at Colorado State was kind of a big part of his research was in trying to quantify the landscape variation and soil concentrations of selenium, and it's definitely variable. Another consequence of the accumulation in these herbaceous perennial plants is that they would actually enrich the soil right around them for selenium because they drop their leaves to the ground each year and they decompose there. So it has the effect of excluding plants that can't handle that concentration of selenium, but it's also been shown to facilitate the growth of other plant species that are tolerant. But it's definitely something that is variable across the landscape, I think.
And we have another question. Bruchid beetle larvae are well known for their discrimination of toxic L cannabinine from L arginine. That's a completely different biochemical arena, but have you noted any variation in foliage/seed predation in the field of the accumulator/non-accumulator?
Yeah, I think the Bruchid beetles, there's got to be a really interesting story in how they have adapted to growing off of these selenium hyperaccumulating plants. Like their seed predation from those is pretty ubiquitous, and these hyperaccumulators are not really excluded from that. And yeah, I think there've been a couple of papers on at least two different ways that insects have avoided the selenium toxicity, and one is just that they don't absorb it through their gut. And the other, let's see, what was that one? It's been a little while since I have looked back at this literature, but I think the other one is that they do something similar to the plants. So the form that these selenium compounds often take in the plants that are accumulating them are modified amino acids that just won't get indiscriminately placed in a protein instead of cysteine or methionine. So I think that, yeah, some of the insects have developed their own biochemical ways of ensuring that they don't get these messed up proteins from the selenium. Yes?
This might be kind of a weird question, but I was wondering if you knew of any substantial sulfur-based defenses in Astragalus species that the glucosinolates and graphene
Mm-hmm. Yeah, I don't know of any toxic compounds that are based off of sulfur, but if you looked closely at one of these plots of the soil to leaf arrows, yeah, like there are all of these samples that have actually decreased the sulfur concentration. So it looks maybe kind of like there's some optimum. And these are growing on some really sulfur-rich soils. And it makes me wonder whether like a potential prerequisite for developing the hyperaccumulation character is an ability to just exclude the sulfur because they're in really high sulfur soils. Okay, if there aren't any more questions, thanks again.